Hello guys, welcome to Dr. Science. So today we are going to talk about how natural killer cells are able to kill the viruses and tumor cells which are present in your body. And not only that, we will talk about the receptors present on your natural killer cells. So this is the image which is taken from your pathology textbook. So I will talk about the image later on, but first we will talk about the functions of natural killer cells. Now basically natural killer cells, the function of the natural killer cells is to destroy mainly virus infected cells, and not only that, they destroy the tumor cells, and not only that, they destroy the cells which are irreversibly stressed or your abnormal cells. Now how natural killer cells are going to do the killing of this virus or the tumor cells or the abnormal cells? Now in order to understand that, we have to know the receptors which are present on the natural killer cell. So suppose this is a natural killer cell, and this natural killer cell, it is having mainly two types of receptors present on it. The first type of receptor is called as your CD16, and the second type of receptor is called as your CD52. Okay, the second type of receptor is called as your CD56, not CD52, it is CD56. Now the function of the CD56 is not yet known, but the function of CD16 is that CD16 acts as a FC receptor. It is an receptor for FC receptor of immunoglobulin G. So what is the meaning of this one? So basically, as you know, suppose there is a bacteria in your body. So this is a bacteria, and this bacteria is having certain antigens present on it. Now during the times of infection, our body will produce antibodies, and suppose this is the antibody that is produced our body, and this antibody name is immunoglobulin G or IgG. Okay, now this IgG basically, so this is IgG, immunoglobulin G, is having two portions. The upper portion is called as your FAB portion, and this portion here is called as your FC portion. Now imagine if there is a bacteria, suppose this is a bacteria, and this bacteria is having antigens, and our body has produced an antibody called as immunoglobulin G. Now the upper part is called as FAB portion, right? And the lower portion is called as your FC portion of the immunoglobulin G. Okay, so eventually the natural killer cells I talked about, they are going to contain a type of receptor called as your CD16, which is a receptor for immunoglobulin G. It means the immunoglobulin G which is present here, this immunoglobulin G, suppose this is the immunoglobulin G and it is having the FC portion. So eventually this immunoglobulin G can go and bind with this CD16 receptor. Okay, because the immunoglobulin G is having the FC portion, right? So eventually, suppose this is a bacteria, I will repeat this again so the basics are clear to you guys. And eventually our body is producing the antibody immunoglobulin G, which is having an FC portion. This is the FC portion. So what will happen? Eventually the natural killer cells which are having CD16 receptor, eventually these natural killer cells will go and bind with the FC portion of the immunoglobulin G. So this is the FC portion, the orange color one is the FC portion of the immunoglobulin G, and eventually the natural killer cells will eliminate this target or the microbe, and this type of phenomenon is called as antibody dependent cell mediated cytotoxicity, or we can simply call as ADCC. Now why it is called as ADCC? Because as you can see here, we are using an immunoglobulin or antibody. IgG is an antibody, and antibodies are going to have an FAB portion and FC portion, and the FC portion is going to attach to the CD16, which CD16 is a receptor for FC portion of the immunoglobulin G. I hope you understand that concept. Now we will go to the other receptors which are very helpful in order to understand how a tumor or a virus cell is going to get killed. So suppose this is a natural killer cell. This natural killer cell is going to have a receptor called as your activating receptor, and this activating receptor is named as your natural killer G2D receptor. Okay, and the other receptor which is present on the natural killer cell is your inhibiting receptor. What is this receptor? This is your inhibiting receptor. So basically, suppose there is a cell. This is a normal cell. Okay, this is a normal cell which is not infected by any bacteria or tumor or any tumors. So this is a normal cell. We will first see what is happening in a normal cell. So basically, when there is a normal cell, okay, suppose this is a normal cell. Okay, my pen is not working. Okay, suppose this is a normal cell. This normal cell is going to express a receptor called as your MHC receptor or MHC class 1 molecule, okay, which is seen in normal cell. Okay, so suppose here is a natural killer cell, and this natural killer cell, it have an inhibiting receptor, right? It is having a, first we will talk about the inhibiting receptor. This is an inhibiting receptor. So whenever there is a normal cell, the MHC molecules are expressed on the normal cells. Whenever the MHC molecules are expressed, the natural killer cell will go and bind with the MHC molecule. Whenever it binds with the MHC receptor or molecule, it will tell the natural kill, or it will inhibit the natural killer cells in killing. It means the MHC molecules are indirectly telling the natural killer cells or activating the inhibiting receptor present on natural killer cells. Hence the natural killer cell will not kill the normal cell. Okay, whereas in the case of a tumor cell, what is going to happen, we will see. So suppose here is a tumor cell. It is a little bit proliferated, and this is a tumor cell. Okay, so this tumor cell, the tumor cell, first of all, it will not have any MHC molecules present on it. It means they don't have any MHC molecules present on it. Every MHC molecule present on a tumor cell is dead. Think like that. Okay, so what will happen, since there are no MHC molecule, there is no one to inhibit the natural killer cell, right? Because suppose this is a natural killer cell. The natural killer cell was having a inhibiting receptor which was inhibited by MHC molecules present on a normal cell. But since a tumor cell is not having any MHC molecules present on it, the natural killer cell inhibiting receptor is not activated. Okay, inhibitory receptor present on the natural killer cell is not activated. Hence there is no inhibition of the natural killer cell. And not only that, tumor cells will express a ligand. This is called as your activating ligand. Okay, this activating ligand will go and activate the receptor present on your natural killer cell, and this receptor is called as your activating receptor. And what is the activating receptor other name? The activating receptor other name is NKG2D receptor. Okay. Once the ligand activating present on the tumor cell activates the activating receptor, aka natural killer G2D receptor present on the natural killer cell, the natural killer cell eventually will eliminate this tumor cell or the virus infected cell. Okay. Now I would like to say some of the interleukins, which are interleukins which will activate your natural killer cells. So suppose there is an infection or there is any virus or there is any tumor, and as you know, during inflammation or during inflammation there are a lot of cytokines produced, right? The main cytokines we will talk about is your interleukin 2, interleukin 15, and let me draw this one clearly. So the interleukin 2, interleukin 15, and other cytokine is your interleukin 12. Okay. So what this interleukin 2 and interleukin 15 will do, we will see. Suppose this is interleukin 2 and this is interleukin 15, and this is a natural killer cell. This natural killer cell is going to have receptors for your interleukin 2 and interleukin 15. So once the interleukin 2 and interleukin 15 goes and binds with the natural killer cell, okay, let me draw this. Okay. So as you can see here, the interleukin 2 and interleukin 15, which are produced during inflammation, are going to bind with the natural killer cell. Natural killer cell is going to get proliferated. Okay. So basically interleukin 2 and interleukin 15 are helping in proliferation of NK cells. Okay. So what is the function of interleukin 12? The function of interleukin 12 is very different. So basically, suppose this is a natural killer cell, and this natural killer cell is going to have a receptor called as your interleukin 12 receptor. Okay. Now during an immune response, interleukin 12 is produced, right? And eventually this interleukin 12 will go and bind with the interleukin 12 receptor present on the natural killer cell, and natural killer cell will produce an interferon called as your interferon gamma. Now what is the function of interferon gamma? Interferon gamma will convert the simple macrophages into super activated. They are like Superman now. Okay. Super activated macrophages. Okay, that is a function of your interferon gamma. So thus when there is super activation of macrophages, they will effectively kill your bacteria or viruses. Okay. So once again, the interleukin 2 and interleukin 15, they are helpful in producing or in proliferation of your NK cells, whereas in the case of interleukin 12, they help in production of interferon gamma by natural killer cells, and this will help in the activation of super activation of macrophages, which will effectively kill your viruses or bacteria. Now let's go to the image which I was showing you guys. So basically, suppose this is a normal cell. Normal cell will be having MHC class 1. Okay, MHC class 1, which is expressed on the surface. Now when MHC class 1 is expressed on the normal cells, this MHC class 1 will bind with the inhibitory receptor present on the natural killer cell, thus inhibiting the natural killer cell in killing of the normal cell. Hence natural killer cell will not get activated, hence no killing. Whereas look at this virus infected cell or any tumor cell. In tumor cell or any virus infected cell, there is no MHC class 1 expression. Okay, virus inhibits the class 1 expression of MHC molecules, and not only that, it will increase the ligand, it will increase the activating ligand. Once there is an activating ligand, it will go and activate the activating receptor, or aka NKG2D receptor present on your natural killer cell. Hence the natural killer cell will get activated and kill the infected cell. So that was about natural killer cells. Thank you guys.